Bicoid is an exciting gene. It was the first morphogenic protein discovered, and it was one of the first genes to be active in a Drosophila embryo, carrying the instructions on the position of the head of the larva. Does it, however, function in other organisms other than fruit flies? Here is a simulation of actual data that you can then interpret. Let's imagine that a DNA fingerprint is done of a number of insects, an advanced fly, a basal fly, a beetle, and a butterfly. And the DNA bands are compared to the DNA segments in Drosophila, which using these endonucleases would include the gene bicoid and Zerk-Nolt. These two are very similar DNA binding protein genes which are active in embryonic development. Then a southern hybridization is performed in which the DNA is transferred to a membrane. In the final step, radioactive probes can be applied which will bind to the bicoid gene to see how widely distributed this gene is in our samples. Also, a probe for Zerk-Nolt is applied, and then a more generalized probe which would bind to both bicoid and Zerk-Nolt, given that they are similar Hox genes which encode DNA binding proteins. How would you interpret these results in which you can observe the presence of the binding of the very specific probes to the Drosophila gene bicoid and the Drosophila gene Zerk-Nolt? These probes bind to DNA for these genes. How would you interpret the distribution of the probe binding the sample? And how would you interpret these results where a probe which generally binds to both bicoid and Zerk-Nolt DNA, given that these two different genes encode similar proteins, homeodomain Hox proteins which bind DNA?